Okay, we come to this chapter called cell division and cell cycle. Coming to these two terms, what do you mean by cell division and what do you mean by cell cycle? We'll talk about cell cycle first. Okay, so what is cell cycle? How do we define it? Cell cycle is defined as the entire sequence of events occurring between one cell division and the next. Okay, over here we see a pictorial representation of the cell division. Alright, the cell which is dividing, let us learn some terminologies. The cell which is dividing is always called a mother cell. Okay, and the cells which are produced, and these two cells which are produced are called the daughter cells. Okay, so in this cell division, this mother cell has been divided to give you two daughter cells. In the subsequent division, in this second round of cell division, this becomes the mother cell, these two become the mother cell, and these four becomes the daughter cell. Okay, so it depends on which cell division you are talking. So it depends who is the mother cell and who is the daughter cell. Okay, coming to what is cell cycle. Cell cycle is defined as the sequence of events occurring between one cell division and the next. Okay, so यहाँ पे this is cell division 1, this is cell division let us talk about this line. This is cell division 2. Okay, so for this cell, this is cell division 1 and this is cell division 2. So the entire sequence of events which are happening between one cell division and the next will be called the cell cycle. Okay, so this entire sequence of similar is for this. Okay, so the entire sequence of events occurring between one cell division and the next is called the cell cycle. Okay, coming to the term cell division. What do you mean by the term cell division? Cell division क्या होता है? Perhaps bio is the only branch of science where multiplication and division is the same thing. Okay, so यहाँ पे भले ही हम लोग बोलते हैं cell divide कर रहा है, but basically we are talking about the multiplication of the cell. The cell multiplies to give you two. It multiplies with 2 to give you 2 cells. यहाँ पे again multiplies with 2 to give you two cells. So in every generation it increases in a geometric ratio, in a geometric progression. Okay, so cell division क्या होता है? Cell division मतलब होता है proliferation of the cell या फिर increase in the number of the cell. तो कैसे increase करता है cells? मान लो अगर एक cell रहा, एक से क्या होगा? 2 बनेगा, है ना? एक से 2 बना, फिर 2 से कितना बना? 4. In every step we can see it is getting multiplied with 2. It is getting multiplied with 2. Okay, and so on. Okay, so this is how the cell division occurs. अच्छा, ये हर cell division में follow होता है? The answer is no. There are different kinds of cell division which follow different kinds of pattern. This cell division is specially for mitosis. Clear? It is specially for mitosis. Mitosis क्या है? We'll come to that a little later. It's a type of cell division, but this is how, generally in the basic sense, this is how the cell divides or multiplies, as you can say. So what happens? Cell का क्या हो रहा है? At the end of the day, the number is getting increased. Initially we had 1 cell and now we have, say, suppose 64 cells. After how many rounds of cell division? After 1, 2, 3, 4, 5, 6 rounds of cell division. है ना? तो after the end of 6th round of cell division we are having 64 cells. So what is happening? Overall we are growing the size of the organism. है ना? We are growing the size of the organism. Now this growth can be of various types. है ना? If suppose if any cut is present on the organism, suppose if we have a cut in our hand, so what do we see? We see a blood clot first, and later on the skin is healed. Okay, so that is called repair. Alright, so repair also occurs. For repair also you need more number of cells to cover that area, to patch it up, है ना? तो यहाँ पे see what are the significances of cell division. Cell division क्या क्या करवाता है? सबसे पहला चीज जो होता है वो है growth. Okay, the size of the organism definitely increases, है ना. This growth can occur in two ways. Either the number of the cells can increase or the size of the cells can increase. We are talking about cell division, so basically we'll talk about the growth in the growth because of the increase in the number of cells. Okay, we come to this word called repair. What do you mean by the term repair? Like I said, if say you have a cut and you want to heal it, so what happens? Your body does it automatically, and the skin which was cut is now repaired. This happens due to, again, rapid cell division at the area of the cut. What do you mean by regeneration? Have you ever seen lizard cut tail when it's cut? You know, later on, after a few days, you'll see a small tail coming out. It will grow in size and finally become up till the size of the original tail. Okay, we can see it with our nails. We can see it with our nails, with our hair. In fact, with our liver as well. Okay, if all the part is cut and only a single, a small patch of that tissue is left, it can also grow back to its normal size. Okay, so this is called regeneration. Regeneration is seen in many animals. Okay, perhaps the best example for you people is the tail of the lizard. Okay, we come to the last significance of cell division, which is reproduction. What happens? Reproduction के लिए भी we require cell division. Basically, many types of cell division. We'll be talking about reproduction as a whole in the later stages of this chapter. Okay, coming to the types of cell division in our syllabus, basically there are three types of cell division. इसका steps भी syllabus में नहीं है, इसका steps भी आपके syllabus में नहीं है. So basically we have only mitosis to study, but let us have a brief overview as to what is amitosis, what is mitosis, and what is meiosis. These are the three types of cell division. What do you mean by amitosis? Amitosis is also called the direct cell division. Okay, it is also called the direct cell division. So what happens in amitosis? Very simply, if this is a cell, this be the nucleolus, this be the nucleus. Fine. When it wants to enter into amitosis, what will happen? A furrowing will take place. A furrowing or cleavage will take place. The cell will be constricted from two sides. It will be constricted from two sides. It will look somewhat like this. And similarly, the nucleus will also be constricted. The nucleus will also be constricted, right? So this constriction will further increase. This constriction will further increase. This constriction will further increase, and finally these two portions will come close to each other, thereby leading to the formation of two individual daughter cells with two individual nucleus. Clear? So this is called the amitosis type of cell division. Where does it occur? It occurs mainly in unicellular organisms, say bacteria and all. Fine. Lower groups of plants, bacteria, lower groups of animals show amitosis, which is the direct cell division. In the indirect method of cell division, we have these two. Okay, people will not ask you what are indirect and direct methods. Just for your own understanding, understand that this direct method involves no mechanism, just a simple furrowing from the two sides of the cell, and the cell gets divided into two. In the indirect method, we have mitosis and meiosis. We'll talk about mitosis and meiosis both over here. The steps of meiosis are not there in your syllabus. The steps of mitosis are there in your syllabus. So what happens in case of mitosis? Mitosis में क्या होता है? Basically, there's this cell, mother cell. It gives rise to two daughter cells. Gives rise to two daughter cells. Of course, in between there are plenty of steps which we'll be discussing later on. Fine. Just for the sake of understanding, please visualize that a single mother cell will give rise to two daughter cells. Okay, I hope you must have seen the genetics and structure of chromosomes video in which I have talked about the term ploidy. Alright, so I'll just give you a brief overview of as to what is ploidy. Have a look. Ploidy. Ploidy means sets of chromosomes in a cell or sets of chromosomes in the nucleus. If one set of chromosomes is present in the nucleus, we call that cell haploid. Clear? If two sets of chromosomes are present in the cell or the nucleus, we call that cell as diploid. Fine, haploid and diploid. Similarly, if we increase and if we make it to 3 sets, what do we have? We have the term triploid. Fine, we will not be dealing with the term triploid in this chapter. We have this in plant physiology, plant fertilization, but we'll be very frequently dealing with the term haploid and diploid. How do we represent haploid? We represent it with a single small n. Diploid को since it's 2, therefore 2n है ना. ट्रिप्लॉइड को अगर करते तो कैसे करते? 3n. Fine. So please remember, henceforth whenever I write n or 2n, it will mean diploid or haploid condition respectively. Remember the number of chromosomes which human beings have. They have 23 pairs है ना. 23 pairs or 46. So वो diploid state में होता है. 23 pairs mean 2 ना. तो इसलिए 2 sets of chromosomes are present in the normal stages of human life, and that is 23. Here 23 pairs is equal to 46. और अगर ये haploid रहता तो क्या रहता? सिर्फ 23, no pairs. Okay, so please mark it or note it down. What is n? What is 2n? And what do you mean by the term ploidy? Okay, so why at all did I give you a lecture on ploidy? Fine, it has a major role to play in differentiating mitosis and meiosis. Basically, in mitosis what happens? A single mother cell which is of 2n type. Now you can very well understand 2n means diploid. Okay, will give rise to 2 daughter cells which will be 2n each. मतलब a single diploid mother cell will give rise to 2 diploid daughter cells. This is the actual definition of mitosis. A single diploid mother cell gives rise to 2 diploid daughter cells. Okay, as you can see very well that the chromosome number between the mother and the daughter cells are the same. The chromosome number in between the mother cell and the daughter cell are same. Okay, hence this is the reason why mitosis is sometimes called equational division or equation division. Okay, this is the reason why it is called as equational division, because there is an equation between the number of chromosomes in between mother cell and the daughter cell. The chromosome number is maintained, it does not change. Clear? And there are 2 daughter cells which are produced. Coming to meiosis, let us see what is the difference in between mitosis and meiosis. In case of meiosis, a single mother cell, this is also of 2n type, gives rise to 4 daughter cells. Gives rise to 4 daughter cells. And all of these 4 daughter cells are haploid. Clear? So what is the major difference? The first difference which we can see is the number of daughter cells. Here it was 2 and here it is 4. The second difference which we can see is the mother cell was 2n and the daughter cells, the mother cell was 2n and the daughter cell were n in case of mitosis. But in case of meiosis, the mother cell is diploid 2n but all the daughter cells are n. Clear? So we can see a reduction. We can see a reduction in the number of chromosomes in between the mother cell and the daughter cell. Basically speaking, it is halved. Okay, the chromosome number is divided by 2. Okay, and hence this is the reason why meiosis is also called as reduction division. It is also called reduction division. Clear? Because there is a reduction in the number of chromosomes during meiosis. Please have a look. Let's recapitulate: amitosis, mitosis and meiosis. Okay, we come to the differences between mitosis and meiosis. I didn't tell you one point, that is, where does mitosis and where does meiosis take place? Of course, they will not take place at the same place at the same time. Okay, mitosis takes place in all the somatic cells or all the somatic organs of the body. Somatic cells or somatic organs mean those cells which have no role to play with the sexual reproduction. Okay, which have no role to play with the sexual reproduction. So cells of your hand, they are somatic. Cells of your brain, cells of your eyes, they are somatic. Cells of your heart, they also somatic. But meiosis occurs at places which especially in the reproductive organs. Okay, meiosis occurs in the cells of the reproductive organs to produce gametes. Now you will tell me, what are gametes? Gametes are sex cells. Gametes are sex cells. Okay, which is sperm in the case of male and ovum in the case of female. Okay, and where will sperm be produced? The sperm will be produced in the testes. So meiosis takes place in the testes. And where will ovum be produced? It will be produced in the ovary. Okay, so meiosis also occurs in the ovary. So testes and ovary, the reproductive organs, and it will produce gametes like sperm and ovum. Clear? This is difference number 1. Difference number 2 is number of daughter cells is 2. Here it's 4. The daughter cells are diploid, matlab the daughter cells are 2n. Yahan pe daughter cells are n. We already know this. Coming to the term cell cycle. What do you mean by cell cycle? Let's recapitulate. The entire sequence of events happening between one cell division and the next is termed as cell cycle. Cell cycle basically comprises of two phases. One is called the interphase and 2 is called the mitotic phase. The interphase can also be called, is also called as resting phase, and mitosis and mitotic phase is called the dividing phase. Okay, though we are calling it resting phase and though we are calling it dividing phase, the meaning is not very clear, because we are calling it resting phase. Okay, interphase is called the resting phase, but the cell actually does not rest. The cell never rests at interphase. Why? Because it is always busy in preparing the proteins, enzymes, cell organelles, DNA, RNA, which are required for the subsequent mitosis, for the subsequent dividing phase. So all the, basically we are calling it resting, but all the materials, all the ingredients, all the raw materials which are required for the subsequent dividing phase is produced during the interphase. Clear? So interphase is not actually the resting phase. If we speak in the true sense, the cell is the most metabolically active during the interphase. The cell basically doubles in size. It produces many types of proteins. The cell organelles are doubled. The DNA should be doubled, है ना? Because we have to give it to the daughter cells. If we look from the previous diagram, we can very well see that here we are having the total number of chromosomes or DNA. Chromosomes के अंदर ही DNA होता है 2n, and here we are giving it individually to the daughter cells as 2n 2n. If we add up 2n plus 2n, it will give us 4n, है ना? So therefore secretly the cell has to divide it with multiply it with 2. Multiply it with 2, तभी ना जाके 4n होगा, and then only the daughter cells can receive equal amounts of DNA. So there is a step called DNA replication also, which happens during the interphase. We'll discuss about them a little later. Okay, so coming to the phases, the steps of interphase. The interphase comprises of 3 steps. What are they? First, growth phase, also called the G1 phase. Clear? Next, synthetic phase, okay, or S phase, and in the end, second growth phase, or G2 phase. Clear? So these are the steps of interphase. Alright, we'll talk about them a little later. Coming to mitotic phase. What happens during mitotic phase? The mitotic phase is again divided into two. Okay, what are they? They are Karyokinesis and Cytokinesis. Karyokinesis and Cytokinesis. Again we have a brief discussion as to what is Karyokinesis and what is Cytokinesis. The term Karyokinesis comes from the word karyon. Karyon means nucleus, and kinesis means division, है ना? So karyokinesis means nucleus का division. Nucleus division is called karyokinesis. Similarly, cyto means cytoplasm, and kinesis means division. So cytokinesis means cytoplasm का division, है ना? So division of a cell, basically if this is a cell, it has the cytoplasm and it has the nucleus. So nucleus has to divide and the cytoplasm also has to divide in order for the overall cell to divide and give rise to 2 cells, है ना? So therefore we have karyokinesis and we have cytokinesis in the dividing phase. So karyokinesis, cytokinesis, we are well acquainted with the terms right now. Karyokinesis of mitosis comprises of certain steps. Have a look at the steps. Okay, so what are the steps? Prophase, metaphase, anaphase, telophase. Yes, you have to remember all of these phases in order. And how will you remember it? You will just remember PMAT, M-A-T, Prophase, metaphase, anaphase and telophase. These are all the phases which will comprise the karyokinesis or nucleus division. Coming to cytokinesis or cytoplasm division. Now this cell division is valid for both for animals and as well as for plant cells. Okay, all of these steps, the interphase, you talk about interphase, you talk about karyokinesis, all of this is more or less same for animal as well as for plant cells. But cytokinesis या cytoplasm का division is different for animals and plants. Okay, for animals we have this mechanism called furrowing or cleavage. Okay, and for plant cells we have for animal cells we have furrowing or cleavage, and for plant cells we have phragmoplast formation or cell plate formation. We'll be discussing about all of them later on. Clear? But let us discuss about the interphase first over here itself. Coming to the 3 steps of interphase. The first step of interphase is called the first growth phase or G1 phase. Okay, the second is called the synthesis phase or synthetic phase, whatever you can call it, and the third is called the G2 phase or the second growth phase. So what happens in the first growth phase? In the first growth phase, basically all the organelles, all the cell organelles, they double in their size, they double in their number, basically not size, and the cell basically doubles in size. Okay, the organelles will double in their number. Okay, and RNA and proteins which are required in cell division will be produced. Okay, as we know, from RNA we get proteins with the help of this process called translation. Clear? So in the first growth phase, organelles will be doubled in their number, and all the proteins, basically all the enzymes which are required for cell division will be produced here. Synthetic phase or synthetic phase is absolutely reserved for DNA replication. The DNA doubles its size in this phase. Coming to G2 phase or the second growth phase, basically the RNA and proteins continue to be produced here, and the second growth phase is basically a checklist. You have all these boxes in which they check whether the specific proteins required for cell division is produced in the correct quantity or not. So basically whenever we are making a list, we make this checklist to be doubly sure whether we are correct in our decision, in our gathering, in gathering the ingredients and everything. So it's almost of that type. Okay, because the cell division should be highly regulated. It is highly regulated and it is very, very systematic, because if the cell division will not be systematic, if it will be random, it will lead to one of the most deadliest diseases, cancer. Okay, so cancer or tumor, malignant tumor as we call it, is nothing but cell division which is uncontrolled. Uncontrolled cell division without differentiation is called malignant tumor. Okay, it leads to the formation of malignant tumor or cancer. So इसीलिए these steps should be highly regulated. It should be very, very systematic. Okay, coming to the mitotic phases, we'll talk about karyokinesis first and then we'll talk about cytokinesis. Coming to stages of karyokinesis in mitosis. The first step which occurs is prophase. Alright, in the normal interphase, the cell looks somewhat like this. Of course, organelles will be present over here, which I have not drawn, which I have skipped deliberately because this chapter basically deals with the nucleus. So we just see a prominent nucleus over here in the interphase. But as it enters into prophase, series of changes takes place. Okay, the first changes or change that will take place during the onset of prophase is condensation of chromatin into chromosomes. What was chromatin? Chromatin was this thread-like structure. The thread-like structure of the chromatin will be condensed to give you specific number of chromosomes. For a human being, the chromosome number is 46. Okay, this is the first thing which we'll see. The thread-like material will no longer be a thread-like material. It will be distinct coiled round short structures called chromosomes. Here we have taken 4. Okay, the second difference or second change you will see is the nucleolus disappears. Third change, nuclear membrane disappears. So as you can see, if the nucleolus is disappearing, if the nuclear membrane is also disappearing, all the constituents of the nucleus will come out into the cell. This is exactly what happens. The constituents of the nucleus, which is now, it's not chromatin, it's chromosome. So chromosomes were there present inside the nucleus. So if the nucleolus is not present and the nuclear membrane is not present, the chromosomes will come out and will swim freely in the cell. Therefore, the chromosomes are free in the cell. This is the diagram of prophase. It's randomly scattered. The chromosomes are randomly scattered. Okay, so this entire structure is called a chromosome. This structure is called, as we know, it's called the centromere. And there are 2 organelles. Since this is a round cell, please remember that round or spherical cells will represent animal cells, and they will not contain a cell wall. So this is the cell membrane, and we know in animal cells we have these structures called centrosome or centrioles. We have 2 structures, 2 organelles called centrosome. The centrosome will have 2 centrioles. You can call it either centrosome or centrioles, your wish. Okay, and they will produce the astral rays. They'll produce the astral rays. These rays are called aster or astral rays. Is this thing clear? So this is the diagram for prophase. Please copy it down. And these are the characteristics of prophase. Okay, another point to be noted is, had this diagram not been of animal cell, had it been of plant cell, how would it look? A plant cell, always make it rectangular. Make it rectangular and make double layer. Double layer, why? The outer layer will represent the cell wall and the inner layer will represent the cell membrane. Okay, and internally all the structures will be the same. Okay, and plus it will not have the centriole. It will not have the centriole. Centriole is not present in the plant cell. In class 9, you know, centriole is absent in case of a plant cell. So a plant cell will not have the centriole at the two poles of the cell. It will only have chromosomes. This is the diagram for prophase. In the subsequent metaphase, anaphase and telophase, we'll only be dealing with the animal cell. You can draw it similarly for plant cell. The plant cell will look somewhat like this. We come to the second phase of karyokinesis of mitosis. What do we see? That the chromosomes which were arranged haphazardly, has now arranged themselves in a specific pattern. Okay, the chromosomes have aligned themselves. Basically in metaphase का एक ही characteristic है, one or two. Basically the first thing which you can see is, before even the alignment, you will notice these, if you imagine this to be the earth, you will notice these longitude like structures. Can you see them? These structures are called the spindle fibers. These structures are called the spindle fibers. They are produced during the metaphase. Basically it begins during the prophase only, but in the prophase we do not show it. We show it in the metaphase. These structures are called the spindle fibers, and at the centromere region of the chromosome, it has a sticky protein. It attaches itself with the spindle fiber. Correct? So the first characteristic of metaphase is the appearance of spindle fiber. Okay. Number 2 is alignment of chromosomes. Okay, the chromosomes are basically aligned. Okay, aligned in a particular region. You can see it can also be aligned here, but it never aligns itself here or here. It aligns right at the middle. Again, if you imagine it to be the earth, this region will be called the equatorial region. So basically it's alignment of chromosomes at the equatorial region. Clear? This entire plate at which metaphase occurs is called the metaphase plate, or otherwise you can call it just the equatorial region of the cell. Coming to the third step of karyokinesis, this step or this stage is called the anaphase. In anaphase, we can see that the chromosome which was in the equatorial region have split. So as we know very well that this chromosome was made of two chromatid. This is chromatid 1. This was chromatid 2. So what happens during anaphase? The chromosome is split. The chromosome is split into two. This part and this part. Okay. So the chromosome will be split into two chromatids. It will be split into two chromatids. So as you can see, one chromatid goes to one corner or one pole of the cell, and the other chromatid goes towards the second pole of the cell. What happens during the twitching, or it is due to the forces of the spindle fiber, movement of the spindle fiber, that these chromatids move towards the two opposite poles of the cell. Similar is for all the other chromosomes. They are split into chromatids. Now they are called chromatids, and they move towards the opposite poles of the cell. This movement of chromosome, from the equatorial region to the pole, basically it's the movement of chromatid from the equatorial region to the pole, is called the anaphasic movement, is known as the anaphasic movement. So what happens, iska characteristics mein what do we see? We see chromosomes split into two chromatids. The chromatids move to the two poles of the cell. And number 3, the movement is called anaphasic move. Okay, so these are the characteristics of anaphase. Please copy down the diagram along with the characteristics. The last step of mitosis called the telophase. So what do we see? What do we see in the telophase? We see the presence of two individual daughter nuclei. We see the presence of two daughter nuclei. What happens basically is during anaphase, the chromosomes which were going, the chromatid rather, which was going to the opposite poles of the cell, they become decondensed. Basically telophase is just the opposite of prophase. Prophase में क्या होता है? प्रोफेज में होता है कंडेंसेशन ऑफ क्रोमैटिन इनटू क्रोमोसोम, है ना, कंडेंसेशन ऑफ क्रोमैटिन इनटू क्रोमोसोम. यहाँ पे क्या होता है? द फर्स्ट स्टेप दैट विल अकर हियर इज डीकंडेंसेशन या डीस्पाइरलाइजेशन ऑफ क्रोमैटिड. नाउ इट्स नो लॉन्गर क्रोमोसोम, है ना, वो स्प्लिट हो गया था क्रोमोसोम एनाफेज में ही. सो वील कॉल इट क्रोमैटिड इनटू what do we get here? Again we get the chromatin. So it's decondensation or despiralization of chromatid into chromatin. This is step number 1, just the opposite of prophase. Prophase का next क्या था? न्यूक्लियोलस disappears, न्यूक्लियर मेम्ब्रेन disappears, है ना. तो इसका भी exactly opposite यहाँ पे होगा. क्या होगा? Number 2, as we can see, the nucleolus reappears. This is the nucleolus. Nucleolus reappears and nuclear membrane also reappears. Hence we can see these two daughter nuclei present. This is the overall happening of the telophase. One more thing, we know that after telophase we'll start with the cytokinesis, and we also know that the cytokinesis takes place differently in animals as well as in plants. All right, so the cytokinesis which takes place in animals involves furrowing. What do you mean by furrowing? Basically I'll discuss it in the next part, but it starts in the telophase itself. What happens in the equatorial region? This is the equatorial region. The cell will be constricted. The cell will become somewhat like this. The cell will become constricted inwards. Alright, the cell will become constricted inwards. This will no longer be there. The cell will become like this. Okay, it will start constricting, and this constriction will further lead, just like amitosis, it will further lead, join, and the cells will separate into two. We'll talk about that later. First copy this. Okay, so let's have a look at the cytokinesis in animal cells and cytokinesis in plant cells. So what happens that in the telophase, let's draw the diagram of the telophase. So in the telophase, we see that the cell was more or less constricted with the two nucleus at the two ends. This constriction will further deepen. This constriction will further deepen. It will deepen from the two ends. This is called the furrow or cleavage. It will deepen to such an extent that two individual cells will be produced at the end of the day. Okay, so this is how the cytokinesis in animal cells progress. Correct? This diagram is important. In case of plant cells, what happens? Let us draw a plant cell. We know this is the cell wall extra in plant cells. Okay, these were the two daughter nuclei present. So what happens basically at the center of the cell? At the center of the cell, the Golgi body and the spindle fibers which were present, it accumulates at the center. It accumulates at the center, and Golgi body starts pouring in secretions from itself. This will further have depositions of cellulose and other carbohydrate to form a structure called phragmoplast, okay, or it is also called the cell plate. This will further, let me draw a bigger diagram, it will further deepen and almost form a cell wall, and hence you will get the two daughter cells, daughter cell 1, daughter cell 2. Okay, coming to the significance of mitosis, as to where mitosis is important, what are the important places where mitosis occurs. So mitosis helps for the growth, as we know, we have already discussed. Mitosis helps in repair, mitosis helps in regeneration, and mitosis also helps in asexual reproduction. Asexual reproduction is seen in animals like yeast, amoeba, etc. So all the unicellular lower groups of organisms undergo reproduction with the help of mitosis. What does meiosis do? Meiosis basically helps in the sexual reproduction of higher plants and animals. It has no role. Meiosis, mind you, has no role in growth. It has no role in repair. It has no role in regeneration, but meiosis has only one role, that is in sexual reproduction. The steps of meiosis are not there in the syllabus, but we'll be dealing with meiosis in just few points and we'll be wrapping the chapter up. So up till now, what do we know about meiosis? The first point we know about meiosis, it occurs in the cells of the reproductive organs. And what does it produce? It produces gametes. Gametes can be either sperm or ovum. Clear? The second point which we know about meiosis is the daughter cells are haploid. Clear? But one major importance of meiosis is recombination. It is recombination, or also called the cross joining. What do you mean by this recombination? What do you mean by cross joining? Basically, we often see that the child which is born to a parent has some qualities of the mother and some qualities of the father. Basically, the 50% of DNA of any child is from the mother and 50% is from the father. And this DNA which is of the baby right now is not exactly 50% of the mother and father respectively, it recombines. Okay, the DNA from the mother and the father, it recombines to give you special sets of DNA, special sets of chromosomes, which is individually of the baby, for the baby itself. This is called variation. Alright, so इसी से पता चलता है that babies of the same parent, suppose siblings, do not look the same. Had the siblings contained 50% of the father and 50% of the mother respectively, they should have looked the same under all generations. Under, like, if suppose a couple is having 5 kids and all 5 are girls, all 5 of them should have looked the same, just like twins. But we see that if suppose a couple is having 5 girls of different ages, they all look differently. This is due to the recombination or the variation which is happening in every generation. Okay, so how does it happen? Let us draw and understand. Basically, if the blue colored chromosome is from the mother, if suppose this is the maternal chromosome, okay, and the red colored chromosome is from the father, or we'll call it the paternal chromosome. Can you see it? Basically, what happens during meiosis? Interchange of the chromatids. What happens if we look at this diagram like this? We can see that one chromatid of the mother and one chromatid of the father are interlinked with each other, are linked with each other. Okay, this X-shaped structure which is formed out here is called the chiasma. This is chiasma formation. It can also be called as synapses. It's also called as synapses. This overall phenomenon is called the synapses. Okay, this is called the synapses. So what happens after the end of synapses? We can see that the maternal chromosome looks somewhat like this, and the paternal chromosome looks somewhat like this. It's containing a blue region from the maternal and it's containing a red region from the paternal. So basically the DNA of these two have been interchanged with each other. Clear? This is called cross joining. Okay, this is called cross joining, which leads to genetic recombination. Clear? This is very important in case of meiosis. One very important point in meiosis. Coming to the last part of this chapter, we discussed the significance of meiosis. The first significance of meiosis is it maintains the chromosome number. Okay, it maintains the chromosome number in each generation. As we know that the chromosome is halved, है ना? The chromosome is halved after every meiosis. So अगर half नहीं होता तो क्या होता? Imagine a parent which was 2n crossed with another parent which was 2n. Okay, अगर meiosis नहीं होता तो इसका gametes में भी 2n क्रोमोसोम नंबर रहता, मतलब इसका स्पर्म और इसका ओवम दोनों में 2n रहता. अगर स्पर्म और ओवम कंबाइन करता फर्टिलाइजेशन के जरिए तो क्या प्रोड्यूस होता? एक जाइगोट विच वुड हैव बीन 4n, है ना? मतलब क्रोमोसोम नंबर उसका कितना हो जाता? 23 मल्टीप्लाइड बाय 4. ओके, इट वुड हैव लुक्ड समवॉट like an alien or something, I don't know, but this would have been a disaster. अब suppose next generation में सोचो ये 4n ये मल्टीप्लाई अगर करता तो क्या प्रोड्यूस होता, एक 8n वाला बेबी. So in every generation the chromosome number would have doubled and the situation would have been a big disaster. It would have been a big disaster अगर meiosis नहीं होता. तो इसको खत्म करने के लिए meiosis क्या करता है, meiosis क्रोमोसोम नंबर को हाफ कर देता है ना. एक 2n क्रोमोसोम से only n comes. This is the sperm, this is the ovum, and they combine to give you again a 2n baby, which is just like the parent. Therefore the chromosome number is maintained, है ना. This is one very good important or very important point of meiosis. The second important point we have already discussed. What was the point? The point was genetic recombination. This genetic recombination takes place during meiosis, and it helps in variation. And evolution. It is very, very important in variation and evolution. So this is importance number 1, chromosome number is halved, and the importance number 2 is genetic recombination. Please copy it down.